Okay, so everyone, next we have Dr. Ben Dubansky. He is with the University of North Texas, and he's going to be talking about a few different things today. First, he's going to give us a summary. So Gary did a great job. Thank you, Steve. I didn't even notice that. Gary did a great job talking about oil and some of the oil characteristics. Now, Ben's going to give us a little background about some of the ways that oil can impact different animals. I'm totally simplifying that there. He's going to do a better job of explaining it. And then he's going to switch gears a little bit and talk about his research. So if you don't mind, after you're done with the biology, can we ask questions? Maybe take a few minutes for questions before you switch into research? Okay, come on up. And your clicker's right there. Literally your clicker.
All right, I'm Ben Dubansky from University of North Texas, as she just introduced me, and I'm going to give a little bit of background. Can everybody hear me? I'm going to give a little bit of background on the biology behind how animals are affected by oil and how animals are able to respond to oiling. So I'm going to start off real simple, and here's a little bit, and then after I give that biology overview of how marine life responds to environmental stress, I'm going to talk about how fish respond to oil in the environment by using my research as an example. So, a little bit about how animals regulate health. We all know that when we get into a hot shower, our temperature doesn't suddenly rise. Right? We're able to regulate our temperature. We're also able to regulate a lot of other different things, different processes, thousands of processes that are going on in the body at the same time. And generally how regulation of temperature works is the same way regulation of temperature works in a house. You have a set point here in the house. We got 20 degrees Celsius. In your body, it's 37 Celsius. And if temperature increases in the room, the air conditioner kicks on until we get back to that set point, and it works the same whether temperature increases or temperature decreases, and then, of course, the heater comes on. So the key concepts that I'm going to talk about in this first part of my talk is one thing that we have to get straight is that animals do exchange materials with the environment. Animals are fairly complex. Like I mentioned, that there are thousands of different processes going on at the same time. Then the other thing is that animals are similar. We all use similar mechanisms to handle similar problems. And animals can detect and respond to the environment, and when things change in the environment, animals have to change and adapt to that change. So the first key concept, animals exchange materials. We're constantly exchanging materials with the environment. We're eating food, producing waste, we're breathing in oxygen, breathing out CO2. And in this schematic, it just shows exactly what I just said. As food comes in, we exchange nutrients into our circulatory system, pump the nutrients around to the different tissues and ultimately the cells. The cells produce waste, it goes back into the blood, we get it out of the blood, and then we get rid of the waste. And the same similar situation goes on. We extract oxygen from the lungs, it goes into the blood, goes into the tissues, CO2 comes back out of the tissues, and then we breathe out the CO2. So, animals are complex. Different animals have unique characteristics that allow them to match to their unique environment. Whether you're a killifish, as a minnow living in shallow waters, or you're a Gulf toadfish, you have very different physical characteristics. But there are similarities that unite all animals. So when you get into a pool, and you've probably all tried this, is very hard to run through the water. But these very different animals, we have a bird, a seal, and a beluga whale, well, they're all shaped in a way that allows them to slip through the water quite easily. But when you get down to the cellular level, we see these same similarities that unite us all. And here is a picture of a kidney. And this is a kidney from a fish. And you can see the individual cells and the collecting tubules where we extract the waste. And it doesn't matter if you're a human or a shark, hope you're not a shark, or a dog, wouldn't be too bad, or a bird, probably a little better. The main point is this kidney of these animals, when you get down to a cellular level, does the same thing. And that's how they're all united. So, the other thing that we do in our cells is that we're able to detect problems, send signals that there is a problem, and then we can fix the problem, or attempt to fix the problem. So, in its most basic way, it works similar to you driving in the car, or me driving in the car last night. When I'm driving around and I see my speedometer and I realize I'm going too fast, so I hit the brake, the car slows down, I check the speedometer again, now I'm going too slow, so I hit the accelerator, car speeds up. So we have our sensor, our stimulus, and our response. So, as a good example, I think, and this actually does relate to oil because it works very similar, is the response to high altitude. Here in Mississippi, in low altitude, we have a lot of oxygen in the air. It's very easy to breathe, and we are acclimated to this high oxygen situation. But let's say we flew to Denver and got off the plane. Now suddenly we're in a low oxygen environment where the air is quite thin, as we all may have heard. We have to change our bodies to adapt to this low oxygen environment. Of course, first when you get off the plane, you're going to breathe more quickly maybe more deeply, to extract more oxygen from the air. But the other thing that happens is a cellular response. And generally what happens, and I'm going to simplify this, is that you produce more red blood cells. So over time, you produce more red blood cells, so you're able to carry more oxygen. And when you go back down here to Mississippi, you fly home from Denver, you still have all these red blood cells. So you're able to actually breathe better. Well, a lot of athletes will go train at high altitudes to get more oxygen into their blood, to have more carrying capacity, get to this situation so that when they go to low altitudes, they're able to handle more exercise. They're able to be stronger and faster, and they probably feel great. Another thing they do is they produce something called EPO, or perhaps if they're at low altitude, they don't feel like going to high altitude, they can just inject red blood cells and then get that higher carrying capacity, like Lance Armstrong did. So, key concepts. As I said, animals exchange materials from the environment, food and oxygen, right? Those are the two big ones. Animals are complex, and different animals match their environment so that they can perform well in their environment. Animals are similar. There's different ways that we're all united. All different animals do a lot of the same things, and animals do respond to their environment, and when the environment changes, they detect the change and they respond to the change, just like going to high altitude. So, my example of having low oxygen, detecting the low oxygen, sending a signal through the body to the different cells to fix that problem by making more red blood cells and EPO, etc., is very similar to the response to oil that happens in much the same way. In fact, the pathways share some key portions that unite the response to low oxygen and the response to PAHs. So I'm not going to get into today, but it's something that is very interesting to me for my future research. So, how this generally works, right? So, just as a breakdown, we start with cells, and this is a little Lego man. And you imagine you put together all these different Legos, and you make some lungs here and some stuff in the gut. I'm not sure what that is, but we got all this other stuff too. To put together those cells as different Legos to make tissues, and then those turn into organs, and then we have organ systems like the digestive system, and then you put it all together and you have an organism. So, cells make tissues, put tissues together, you have organs. Put organs together, you have an organ system. Put it all together, you have an organism. So, as I mentioned, animals have many similarities, but the laws of nature apply to all organisms. Organisms live in different environments, but they use similar strategies to attack the same problem. They use similar strategies to deal with oil. So, before I move on, are there any biological questions? Sure. Does anybody on the line want to chat in their questions if you'd like? I'm going to give an example here first. All right. So here's an example of how it works similarly. Imagine a little fish gets eaten by a big fish, but the little fish is soaked in oil. So if you focus in on this little part of the gut, and you imagine that, this is the gut, this is the intestine, it's a tissue, it's made of cells, individual cells here. Oil is going to go through the cells. It's going to want to go right through the cells and get into the body up here. But we have mechanisms that stop the oil or that help try to stop the oil, convert the oil to a more usable product, something that we can get rid of to detoxify. But how do we do this? How do we stop the oil? First, oil is detected in the cell. Then a set of instructions is issued, and hopefully the instructions are followed to the best of the organism's ability. So every cell has the same instruction manual. In every cell, you have the same set of DNA. You have the same genome, right? We call the instruction manual the genome, but each instruction manual is made of individual chapters that we call genes. And each chapter tells the cell how to make the materials to function, specifically for that cell. So what genes from the genome does a cell need to detect the oil? There's a set for that. What genes do they need to send a signal? And what genes do they need to fix the problem? Well, the genome is huge. There's tens of thousands of genes, and these genes are used in combination. So that set of instructions is very complex. And there's 8 million possible combinations in one animal. Well, if you put two animals together from one sperm and one egg, you have 70 trillion possible combinations. Imagine that. But what I want you to understand is it's not just an on-off switch. These genes function in sets where you could have a pattern of on-off. And the on-off isn't just an on-off switch. It's more like a throttle, right? So you could throttle it up and you can increase the power, or you could throttle it back and slowly decrease the power of each gene, working kind of like this set of dip switches. Well, animals can alter the throttling, the individual gene throttling of every gene, and clusters of genes can be coordinated. So you can imagine that set of genes used to detect the signal can be slightly altered when need be. All right. The other thing is there's a known pattern for any given situation, and once we figure out that pattern, we find out that it's predictable and it's common to many different animals. So when we're exposed to oil, or when a fish is exposed to oil, the pattern is very, very similar. So here's how it works. Here's just an example. These are breast cancer genes. And here we have 6 animals in each one of these rows, and it's a lot of genes, I know. But if you imagine that the brighter the green, the more upregulated the gene, the more throttled up the gene is, or the brighter the red, the more downregulated or throttled down the gene is, you can look at these 6 different animals and note that in 4, 5, and 6, you have a lot of downregulation of these genes, and in 1, 2, and 3, you have a lot of upregulation of genes in these different animals. And obviously 1, 2, and 3, or 4, 5, and 6 have different states of their genes, right? So that predictable pattern is set for the 3 of the animals, so we can detect what this pattern is for breast cancer. All right, how it works with oil. So here's a heat map of thousands of genes. This is a lot more genes than I just showed you for breast cancer, right? This is a whole animal. But there is a pattern, and we're able to find that pattern and use computers to show us what the pattern is and figure out the statistics behind it to say whether or not the pattern is indicative of oil or not, the exposure to oil. So the pattern is known, it's very predictable, it's similar in many, many animals, and it tells us about what's going on inside the cell and the rest of the body, depending on what tissue it came from. So, any questions about the general biology? Feel free to comment on the internet. Yes?
Does that have an epigenetic effect that might be
It's funny you should ask. So the question is if during spawning season, or actually at any point in time, if an animal is exposed to oil, does it have an epigenetic effect? So an epigenetic effect is what is not encoded into the genome, but rather how the gene is altered in a way that changes the expression of the gene. And sometimes, and we're finding this in our lab, and this is published, that if you expose an animal to a stressor, even though you're not changing the sequence of the genes, you alter the gene expression in the next generation. And we've seen this in animals exposed to PAHs as well. Zebrafish, there's a nice study that came out about zebrafish, and they exposed them to single PAHs and showed that, yes, in fact, it does pop up. And the other thing that's interesting is it washes out over successive generations slowly, and sometimes 2 generations later, something pops up that's not encoded in the genome, but rather in control of the expression of the genes, in control of the pattern that I showed you back here. So epigenetics is an interesting and it's a tough thing that is very difficult for scientists to understand, and is part of our research program with the Gulf oil spill presently. And with some other funding too for something totally different, but still epigenetics. In fact, it's hypoxia, low oxygen. So the two do come together. Any other questions? Okay, so on another statement, sometimes it's good to use just males or just females, especially during the breeding season, like what happened in the oil. So we just use males in this case. Try to be careful with what could be happening during the reproductive cycle. So with my research started before the oil made landfall. So we got out there very quickly. We went through Louisiana, Mississippi, and Alabama and sampled numerous sites. We collected killifish, Gulf killifish, I'll talk about them in a second, and sediments and water from Barataria Bay, Louisiana, and in the other 2 states. And we've actually been to dozens of sites that we've visited and sampled over 3 years, and then we're going back to now as well. So just a brief overview of our results and just observations from field lab experiments where we took stuff from the field, fish, oil, sediments, brought them into the lab, did experiments with the stuff. Fish, what we found in the field is that fish showed sign of exposure long after you saw visible oil. There was still oil in the sediments. And then gene expression is significantly altered at oiled sites, as you might imagine, in these fish. And sediments, exposure to sediments in the lab caused developmental deformities in embryonic fish, and I'll show you some of that in a minute. And that because of this and because of the prolonged effects that we're seeing, we believe that long-term monitoring of sites like this is potentially necessary to understand the recovery of the Gulf. So we did go out and we trapped a lot of fish, Gulf killifish. Here's locations of the early field sites that we went to, Grand Terre, and again I said I went to dozens of sites in Barataria Bay, a couple sites in Mississippi and 3 sites in Alabama. We measured the signal. We looked at those first 6 sites in 2010 and 2011, looked at the Gulf killifish before and after oil made landfall. In the field, we looked at the adults, and in the lab, we looked at the effects of the embryos of these Gulf killifish. Now, you might be wondering why we care about these little Gulf killifish. Well, Gulf killifish are a great sentinel species to look at for understanding the effects of the environment on fish species. Well, for one, they're widely distributed. They're all the way across the Gulf. I go down in Texas and collect Gulf killifish now. They're all the way over into Florida. They have a very limited home range. We have excellent background information available. The genome is sequenced. It's an excellent wet lab rat. So we have a lot of molecular tools that we can use to study these fish. They're obviously important in the food web because they're bait fish. They have rapid development and large transparent eggs, which makes them very easy to view under the microscope. But this limited home range thing is great because these fish don't migrate. They stay in the same place their entire life. So we can grab some Gulf killifish at a specific site and find a site-specific indication of what animals are doing at that site. And that's one of the big things that I like to say about Gulf killifish is to drive home that point, is they're great fish to use in field-based experiments. And the other reason is for lab-based experiments. So, I'm not going to go into this too much. I just want to break down here is part of the signal. And here we have upregulated genes in yellow, downregulated genes in blue. This is the first trip we took, pre-oil, the red line is oil, second trip, third trip, fourth trip, again, first, second, and third, first, second, and third, just to make this graph smaller. And then we normalized it to a 0, to a control fish, which is this black. We normalized these next 3 trips to pre-oil conditions. And what we saw is that, as you can see in this yellow, at these oiled sites, the gene expression pattern looked wildly different than at these unoiled sites, and it followed a pattern. So we see a divergent gene expression pattern, and here, in this example, this is Grand Terre, which is coincident with the location and the timing of the oil in these fish. So if you measure the pattern, and you're looking at gills versus liver, and I've said before that each tissue in each cell could do a different pattern, right? Because imagine what the tissue or the organ is made for. So the gill and the liver do very different things. So if you look at the gill and you look at the liver, I'm not going to click on individual genes, don't worry. You can see that there's a difference in the expression pattern in the gill and the liver. And you also see there's an expression pattern between Grand Terre, our heavily oiled site, and then Bay St. Louis and Bayou La Battery here. And then if you look at both of them, what you see is although there's differences in the pattern of gene expression in the liver and the gill, that pattern converged, and there you see the similarities I was talking about. So even within an organism, there's similarities. We have the same genome throughout all of our cells, and certain key genes are on or off in response to something like oil. So we see that the genes that are throttled up in those 100 genes that both tissues share have to do with stress, the immune system, and sex hormone responses. We also see the throttled down genes have to do with feminism, and these are all males, right? So we see that some of these genes are involved, and we see this in exposure to toxicants and other situations too, is feminism of males. And this is a complicated figure here, and it shows the interaction networks of that huge pattern. So if you go back to that other slide with all those crazy dots all over it, and you try to unite all of the genes that are turned on and off around similarities, they're all united by this thing called the aryl hydrocarbon receptor pathway. Many of them are, at least. And then you see the genes between the gill and the liver. Here's the shared ones. And these are the biological pathways that are controlled by these genes as we know it. So in a whole body pattern, we see that sex hormones are altered, stress, immunity, response to wounding, inflammation, dermatological disease, immunological disease, of course cancer, and that in our data we found that these changes in the gill were much more dramatic, and that might make sense because the gills are constantly being exposed to the water, right? So, detect, signal, and fix. And someone's going to talk about this in a minute, but generally what happens is the oil comes into the cell, a receptor called the aryl hydrocarbon receptor detects the oil. It comes into the nucleus, and a signal is generated by producing gene transcription. It turns on and turns off these genes, and these genes are produced, and we can measure it and figure out what's happening. What is the cell doing to fix the animal? And this is just the beginning. So part of those patterns of genes, so part of that group of genes I was talking about, are unified around the aryl hydrocarbon receptor. And what happens is when the aryl hydrocarbon receptor transcribes genes, we have a known pattern, something that we know very, very well. In fact, the aryl hydrocarbon receptor pathway is perhaps the most known soluble receptor pathway in science. It's an archetype for other cells, or other pathways, excuse me. And one of the stars that gets produced is called CYP1A. Well, CYP1A, it's a very popular bioindicator of oil exposure. We use this protein, we measure this protein, and it's very predictable that when animals are exposed, we'll see CYP1A. Well, just going back a little bit, at our oiled site, and I need to get this out, we found that in the water we really did not find a lot of elevated hydrocarbons. We didn't find a lot of toxicity in the water at our sites, except at Grand Terre in June, but it was really barely above detectable limits. So I need to disclose that. In the tissues, when we measured the tissues, the muscle, just like the seafood testing industry, we really didn't find any PAHs in our fish. I mean, really, no site really stood out. We found a little bit, but very little. And in the sediment, the oil, the PAHs in the oil were elevated for a very long time. And what we found was these fish expressed a lot of CYP1A, that indicator of exposure to oil. So although they showed signs of exposure to oil, we still didn't find oil in the tissues. So if you look at a liver, and you're looking for CYP1A, and you look between these oiled sites and reference sites, and this is just an example, you see in red, this is CYP1A, is this red color. You see more at an oiled site. And we can test the molecule too. We can measure how much CYP1A is in the animal. And we find, and this is, we have a sample size of 84 here, so from 84 fish from the control, 84 fish from the reference sites, we see elevated CYP1A in all these fish. And here's a picture of a gill, and I've talked before that the gill transcriptional response was a little bit more intense than even the liver. So in red here is again CYP1A, and if you compare a reference site to an oiled site, you see a lot more CYP1A, and you see things like aneurysms and a little bit of swelling in the gill lamellae. Here's another picture. You see more swelling in this fish, and you still see that stark contrast. But overall, if you compare the oiled sites versus the unoiled sites, again, we didn't find a lot of PAHs in the oil. We didn't find a lot in the tissues. But we found CYP1A indicating that these animals were responding to oil. probably in the sediments, right? So we find it in the gills, the intestines, the head kidney, and the liver. And the head kidney is we'll just call it the kidney for fish, simplify things. And you compare oiled sites versus unoiled sites. So the next thing we did is we took the sediments that we had collected, sediments that apparently had a lot of oil in them, and we tested to find out how much oil was in the sediments, and we spawned killifish and exposed them to these sediments. We put them on top of the sediments. And we looked to see how these animals hatched and how they developed. Well, the first thing is there was a lot of CYP1A in the larval fish. So when we cut these fish up, we see a lot of CYP1A. This is a larval gill. You're just going to have to trust me on that. You see a lot more CYP1A in the gill. You don't see a lot in the intestine, because at this point, obviously, the fish aren't eating, really. They're either utilizing their yolk. But you see a lot in the liver, of course, and a lot in the kidney. So here's a little schematic we have. Interestingly, this is an unoiled site, and this is a lightly oiled site right here, where we still see indication of CYP1A, but we didn't see any problems with the embryos. So the embryos seem to develop just fine, but they were exposed to oil. But the animals that didn't develop just fine from the heavily oiled sediments, the ones that were developed on heavily oiled sediments, had an elongated heart and pericardial edema, which you may have heard about. You know, you may have a relative that had pericardial edema. It's just a buildup of fluid in the heart cavity where the heart cavity swells, and then you have this really elongated heart. It doesn't quite look like a heart anymore. The other thing we found is a lot of tail hemorrhaging. Fish pump blood back to the tail, and it's got to come back up. So if the heart's not pumping well, obviously blood's going to pool. We're going to have some issues, perhaps thrombosis or whatever. So we see these morphological defects, failure to hatch, slow growth, and then the ones that do hatch from heavily oiled sediment exposures kind of look like this. And they're generally listless and not capable of feeding, and they die. So it's pretty obvious contrast. So in conclusion, so organisms exposed to oil show this characteristic genomic pattern where we see indications that they're detecting, signaling, and fixing the problem, or attempting to fix the problem, and that organisms from oiled environments had indications that they had reduced reproductive capacity. And I showed you this slide earlier. This showed signs of exposure long after visible oiling was apparent. Gene expression altered. Sediments cause developmental deformities, and long-term monitoring is definitely necessary. So, you know, this is a basic pie chart, and you can imagine that an animal, you know, you devote a certain amount to general metabolism, a certain amount to activity, a bit to growth, a bit to reproduction. And there's other pieces of this pie, too, that aren't shown here, like immunity. Well, we all know that when you get sick, like I am right now, there's a metabolic cost associated with it. The immunity part of the pie expands a little bit, and it takes a toll somewhere in here with these other portions of the pie, right? So you think about the energy budget of a fish, and the fish has to devote a certain amount of energy to dealing with oil, and oil causes all these other problems, like immunology, problems with the immunology of the animal, immunity, reproductive output, whatever. The portions of this pie are changing, causing the animal to, you know, undergo stress, and there's likely a metabolic cost associated with this. So what we're doing now is I'm working with the Recover Consortium, funded by Gomery, and we're looking mostly at mahi and redfish in the Gulf, these two economically important species, and we're looking at the cardiac effects of exposure to oil, the sublethal concentrations of oil. So in the past, we've had this paradigm of where we expose animals and we count how many die, and we call that kill them and count them. And that's the most popular way to do things, and that will never go away. We have to know what the toxicity of oil is. But the other thing is that animals, as I've shown you, can recover. They can respond to the oil. But there may be a metabolic cost associated with that, and we are seeing that that is in fact the case. So we're looking at these fish, and we're also looking at birds. And in particular, we're looking at bird embryos, the embryonic development of birds, and how oil affects development in birds. In particular, how it affects the heart. And of course, I'm not going to go away from my killifish, who I love so much. So a little bit of acknowledgements. Again, most of this work was funded by the Gulf of Mexico Research Initiative, or GOMRI, which I like to call GOMRI, sorry, and the National Institutes of Health, NSF, and then some of this was funded by the Multi-District Litigation Committee for the civil case a little while back. Again, Andrew Whitehead, we did a lot of work together, and Charlie Rice from Clemson, and then of course Fernando Galvez, my PhD advisor, was instrumental in this, and there he is. And then Warren Berger, and I don't have a picture of him here, but he's who I'm with now with GOMRI, and that's it. So if there's any questions, I should also add that a lot of other people took part in this. Les Barrios, Captain Barrios, got us all over wherever we needed to go and lent us boats when we needed them, and a few other people too helped quite a bit. So if there's any questions?